Hey there, welcome to Chapter 27, The Protists! Woohoo! Okay, by the end of this chapter, you should be able to define what a protist is, as well as anyone can, and then explain why protists are considered a paraphyletic group, summarize the ecological and medical importance of protists, explained how endosymbiosis played a role in the origin of mitochondria and chloroplasts, describe the diversity of feeding, locomotion, and reproduction in protists. All right, so we've already talked about the prokaryotes, the domain archaea and domain bacteria. Now we're going to talk about domain eukarya from here on out. So this is the third domain on the tree of life. There are features that distinguish eukaryotes from bacteria and archaea. Most eukaryotes are large. Most of them have organelles and a more extensive cytoskeleton. They have a nuclear envelope, right? Their nucleus is enclosed in an envelope. They have, some of them, but not all of them, multicellularity. They can reproduce sexually or asexually. And so today we're going to talk about the protists. The protists include all of the eukaryotes that we don't call land plants, fungi, and animals, okay? So if you're wondering if this was a monophyletic group, hearing that they're everything except this, that, or the other is kind of a clue that maybe they're not. Some examples of prokaryotic diversity. If you look in the open ocean, you can see all of these microscopic protists like diatoms with their beautiful glass shells. Looking in the coastal waters, you can see enormous protists like kelp that form the structure for underwater forests that other animals and things live in. And you can look in the intertidal habitat, and you can see brown algae on the intertidal, making a place for animals to live. All right, so why do we care about protists? First, they have ecological importance, which I'll get into. Second, if you don't understand protists, you can't really understand where plants, animals, and fungi come from. Third, there are protists that have an effect on human health and welfare. So just some examples of protists that we should care about from like our own selfish human perspective. The Irish potato famine of 1845 was caused by a protist, and that potato famine killed the potatoes, and that led to mass immigration and starvation. So malaria are also caused by a protist. So malaria is a chronic public health problem that occurs worldwide. There are 5 species of a parasitic protist called Plasmodium that we get from mosquitoes. So let's start over here. So the mosquito will bite somebody, and let's see, inside the gut wall, the mosquito will bite somebody, and the Plasmodium cell types that are in the human blood will infect the mosquitoes, and so the mosquito will pick up the male gametes and the female gametes, and these will go into the wall of the gut of the mosquito where it's storing all of the blood that it just ate, and so the sperm and the egg will come together, be fertilized. It'll form a zygote plasmodium that will undergo meiosis, followed by mitosis, so now you have a whole bunch of them. And then they grow up, and then they're kind of at this stage when the mosquito bites the next person, and so those little bitty plasmodia will go into a person through the next bite. They'll work its way into the liver cells and then reproduce and reproduce and reproduce by mitosis, eventually exploding out of the liver cell. They then go into the red blood cells and reproduce by mitosis within the red blood cells, and then they're passed on to somebody else. And so clearly malaria is responsible for much death and disease worldwide. Let's see, here's a little picture of a malaria parasite invading the red blood cell, so it's this stage right here. So you can see it's getting lumpy with all the plasmodia until it eventually kind of explodes and releases all the plasmodia out into the bloodstream. All right, so Plasmodium is still out of control. We cannot control Plasmodium, so malaria is not under control at all. People have tried to come up with drugs that control the growth of Plasmodium, but Plasmodium has evolved resistance to those drugs. It evolved so quickly that researchers have not been able to develop a vaccine against it. People have tried to use insecticides to control the mosquitoes that spread malaria. However, over time, the mosquitoes have evolved resistance to the insecticides by natural selection, so that's not working very well either. All right, here's some other health problems caused by protists. I don't know why I put these people at the top, but this is kind of how these diseases make me feel. Malaria, as I said, brain-eating amoeba, so that's another protist that sounds great. The infections with this are almost always fatal. Toxoplasmosis causes eye and brain damage. We're going to talk a little bit about harmful algal blooms. Giardia, so that's one. If you drink still water, in like a puddle or something when you're camping, you'll get severe diarrhea. Trichomonas vaginalis is a reproductive infection, can cause infertility. All sorts of other things, sleeping sickness, etc., amoebic dysentery. So, if you didn't think protists were important, now you know. All right, so periodically in some places on the coast, you will get harmful algal blooms. These are commonly known as red tides. So they look like this or like this. And if you look at them up close, you'll see that the redness is made up of these little tiny protists called dinoflagellates. They are harmful to humans because these dinoflagellates are producing a little tiny bit of toxin. And a lot of the other animals don't care about the toxin. So for example, clams and shellfish are delighted to have all of these dinoflagellates to eat, but humans can be poisoned if humans eat the clams and shellfish that have eaten the dinoflagellates. Protists are extremely important in aquatic food chains, so they represent just 10% of the named eukaryotic species, but they are extremely abundant. We just tend not to give them names, right? They are primary producers, and we'll talk more about primary producers when we talk about ecology, but I'll just tell you that they're species that produce chemical energy by photosynthesis, and if you look up where all the carbon dioxide that is fixed on Earth is coming from, most of it is due to marine protists. We have a common name, plankton, which refers to diatoms and other small organisms that drift in the open oceans or lakes. So plankton is kind of like a generic catch-all term for lots of different kinds of organisms, including protists. And so protists tend to be at the base of the food chain in aquatic environments, right? So they are the things that are photosynthesizing and then getting eaten by other things. So people have thought a lot about whether protists could help limit global climate change. And so we'll talk about the global carbon cycle. This is the movement of carbon atoms from the atmosphere to the organism and then back to the atmosphere. And so this is kind of what happens in the ocean. So you have carbon dioxide in the atmosphere, and these primary producers, different kinds of protists and other things, are undergoing photosynthesis, and they're using this carbon dioxide. They're then getting eaten by primary consumers, like slightly larger things, like this could be a protist like an amoeba, or something like an animal, like a snail. And then these things will die and fall down into the bottom of the ocean, where they'll get scavenged by scavengers, and then the producers and the scavengers will die, and then they will sink down into the bottom of the ocean. So a lot of these creatures have shells made of calcium carbonate. You know what that is? It's like chalk. Yeah, kind of like chalk. Yeah. It's kind of like a white chalky shell, like a clam. So all of those things, all of those like shells and stuff and cells will sink down into the bottom of the ocean, and then they just stay there, okay? So they just kind of accumulate. And so you have this huge amount of carbon stored up in the bottom of the ocean in the form of cells and shells, okay? So this is called a carbon sink, and because it's just a place where you end up with an accumulation of extra carbon. So one thought that people have had is to try to reduce global climate change by decreasing carbon dioxide concentrations in the atmosphere and increasing the amount of carbon that's stored in aquatic and terrestrial environments. Like I said, protists act as carbon sinks, and so they're like this repository of carbon, both the living ones and the dead ones. There are sedimentary rocks, in the oceans that are made out of protists with calcium carbonate shells. Also, the kind of crushed accumulation of bacteria, archaea, and protists on the ocean bottom has become petroleum, right? And so one thing that people have suggested is that maybe we can speed up the carbon cycle by fertilizing the ocean with iron And so the thought is that that if you do that, then the number of protists are going to increase because they have a little bit more iron, and then the carbon that will be kind of pulled out of the atmosphere and kind of tied up on the bottom of the ocean. And so that's just one thought of one way to get carbon, excess carbon out of the atmosphere and lock it up in the bottom of the ocean. All right, so what's a monophyletic clade and what's a paraphyletic clade? All right, Bird, do you remember what a monophyletic clade is?
Yeah, monophyletic clade is like an ancestor and all of its descendants.
Yeah, it's an ancestor. It's a group that's an ancestor and all of its descendants. Does anyone remember what a paraphyletic clade is?
Is it an artificial grouping of like organisms that are related?
No, that's not a paraphyletic clade. That's a polyphyletic clade. Does anyone remember what a paraphyletic clade is? Yeah, it's when it's like an ancestor and some of its descendants, but not all of them. Yeah, so it's an ancestor, and some of its descendants, but not all of its descendants. Okay, so remember what a paraphyletic clade is, and then let's talk about protists. So remember we talked about monophyletic clades are defined by their synapomorphies, right? So what's a synapomorphy?
It says it right there on the slide.
Yeah, it does. It's a trait that's unique to a monophyletic group, right? All right, protists are not a monophyletic group. Protists are a paraphyletic group. Protists do not have any synapomorphies, so they don't have any traits in common, and they're not monophyletic. And if you don't believe it, look at the DNA sequences, and you can see that if you do a phylogenetic analysis, we find that this is true. One name that we used to kind of, back in the older days, talk about when you have a group that is just kind of a random assemblage of things, we'd call that a garbage can group. So I would say that protists are kind of a garbage can group because they're everything except X, Y, and Z, right? So they're paraphyletic. All right, so we do within the protists, though, there are groups of protists that do form monophyletic groups, and some of these can be categorized according to distinctive synapomorphies or distinct, structures or organelles. So there's one group of protists called the Stramenopils, and they have these weird flagella that have hollow hair-like structures over the outside and like branches on the hairs, like crazy stuff, okay? So that's something the Stramenopils have that nothing else has. There are 7 major groups of eukaryotes that have been identified on the basis of their diagnostic morphological characteristics. So there are the Amoebozoa. We'll look at some of these in a bit. They don't have cell walls. They can kind of ooze around in these weird lobes, Excavata. So these have reproductive cells with one flagellum at the base. So we'll look at this. This lineage, though, is a lineage that gives rise to the fungi and the animals. Okay, so I'll show you a picture of that lineage in a little bit. So this lineage isn't monophyletic, but Amoebozoa are. Excavata are, I think, also monophyletic. So most of the cells have a feeding groove, and so I'll show you a picture of that. And they don't have mitochondria. The Plantae, you know them plants, right? So that is kind of nested within the protists. So I'll show you that on the tree in a minute. Rhizaria. The cells lack cell walls, and they have these cute shells, which I'll show you. The alveolata, kind of like the alveoli in the lungs that form these little sacs. They have cells with sac-like structures, but I don't think we can actually see that. Just take my word for it. And then the Stramenopila, which we already showed you, have those weird flagella with the hair-like projections that branch. All right, so those are the 7 major groups of protists and other things that come off of protists. All right, so here is our best estimate for the phylogeny of the protists, okay? So morphological data and DNA sequence data together suggest that the 7 groups are monophyletic. So the protists aren't monophyletic, but each of these groups is monophyletic, okay? That is, if you're willing to consider fungi and animals to be part of the Opisthokonta. If you're cool with that, that's monophyletic. If not, then I guess it's not monophyletic. Okay, so let's see here. So within the protists, so you can see the 7 groups, some of the groups are sister groups to each other. So for example, the Amoebozoa and the Episthoconta, you can see this yellow and this red are more closely related to each other than to any of the other protists, right? So they're kind of sister groups to each other. So this whole lump here could be a monophyletic group called Uniconta, okay? And then the other 5 lineages are, the rest of them, right? So those also form a clade. This would be the ancestor and all of its descendants, okay? That's biconta, two-tailed. And so one hypothesis suggested there was some split between the uniconts and the biconts a while ago. All right, so how are protists paraphyletic?
Oh, it's just because, like, animals and fungi aren't really called protists, and then they kind of come out of the protists.
Yeah, it's not just animals and fungi. What's the other thing that we don't usually consider to be a protist?
The plants! The land plants!
Yeah, the land plants. We don't usually call those protists, right? We call those plants. And they seem to come out of the one of the lineages of protists, right? And fungi and animals come out of the Episthoconta lineage. All right. I think we mentioned this part. Which lineages does the name Protist exclude? We already said it. Fungi, animals, and land plants. Okay. Before I start this, I just want to make sure everyone remembers, what is environmental sequencing?
Oh, I know. That's when you take some dirt and you just, like, extract the DNA from it and then see what the sequences of DNA are and then match them up on the computer, the DNA, where you know what it is.
Yeah, that is environmental sequencing. Okay? So we have discovered new lineages of protists using environmental sequencing just by taking samples of water or air and then figuring out where these organisms go on the phylogenetic tree. So we think that the earliest eukaryotes were probably single-celled, and they had mitochondria, a nucleus, an endomembrane system, right? Remember what an endomembrane system is? What goes into the endomembrane system?
The Golgi apparatus!
Yeah, that's part of it. The Golgi apparatus. What else?
The rough endoplasmic reticulum.
Yeah, the rough endoplasmic reticulum. Yep. They also have a cytoskeleton, and there's no cell wall, right?
Wait, how come they don't just ooze all over the place?
Well, because they still have a cell membrane, right?
Oh, yeah.
They're just missing a wall, like a rigid wall of cellulose. We think that the earliest eukaryotes probably swam around using a novel type of flagellum, so this is a euglena. What I mean by novel is bacteria have flagella, but protists, and animals actually, have a completely different kind of flagellum. So it's a more complicated kind of flagellum that eukaryotes have. All right, so protists are considered a paraphyletic group because why?
That's hard.
All right, well, why don't I say some and you say if that's the reason or not. Okay, so is this true? Protists are considered a paraphyletic group because only some of their traits are found solely in protists. Other traits are also found in other organisms.
I mean, that can't be it. That happens all the time with monophyletic groups, too.
Yeah. Protists are considered a paraphyletic group because they can be unicellular or multicellular.
That's not it.
All right, protists are considered a paraphyletic group because they share a common ancestor.
I mean, if they share a common ancestor, they could be monophyletic or paraphyletic.
That's true. Protists are considered a paraphyletic group because they are not defined by any synapomorphies.
Oh, I kind of like that one, but I want to hear the last one.
All right, protists are considered a paraphyletic group because they represent all of the descendants of a single common ancestor.
Oh no, if they were that, then they'd be monophyletic.
Okay, so what's the right answer?
It's D.
All right. So let's talk about how all of those protists got them organelles in them. okay? So let's just start with mitochondria. So those are the organelles inside eukaryotic cells that generate ATP. So the endosymbiosis theory is that the mitochondria happened when a bacterial cell took up residence inside another cell, and we think that this happened about 2 billion years ago. So you have a host cell, and it's like, bacterium. So this is a bacterium, and this is a bacterium, and the outer one goes to engulf the inner one, okay? And instead of digesting it, it like allows the bacterium to live inside the host cell, and we can see a couple things. First of all, we can see that this cell inside the larger cell has two membranes, right? The membrane that it already had before it came in, and then the membrane that the host gave it when it engulfed it, okay? And so they live in a symbiosis. A symbiosis is when individuals of two different species live in physical contact. And actually, this definition isn't quite complete. You have a symbiosis when individuals are living in some kind of relationship, but an endosymbiosis is when the organism of one species lives inside the cells of an organism of another species. So this is an endosymbiosis, okay? And in this kind of an arrangement, the host is able to give the bacterium protection from the outside world and also give it the carbon compounds that it needs, and the bacterium is able to supply the host cell with ATP. And we have evidence that this happened, other than, it sounds like a pretty reasonable idea, but we actually can demonstrate evidence that it seems to be true. All right, so mitochondria are about the same size as a particular kind of a bacteria. Oh, look, it made it a bacteria. This is supposed to be an alpha. Alpha, like that, alpha proteobacteria, okay? And mitochondria, like bacteria, replicate by fission. What's fission?
That's when they divide in half.
Yeah, that's when they divide in half. If you look at mitochondria, they have their own ribosomes. What are ribosomes?
Oh, those are like those little snowmen that are on the endoplasmic reticulum, and they kind of go in there and they like take the messenger RNA and they use that to make amino acid chains.
Yeah. So mitochondria have their own ribosomes and manufacture their own proteins without the help of the nucleus or the ribosomes that are in the rough endoplasmic reticulum. All right, mitochondria have double membranes, right? Which I just showed you. You would expect they'd still have the membrane they came in with and the membrane they're coated with from the host. Mitochondria have their own genomes, so they have circular molecules, just like bacteria have circular chromosomes. And mitochondrial gene sequences are more similar to the alpha proteobacteria sequences than the eukaryote nuclear DNA sequences. So let me make sure that makes sense. You have a host cell, you have a mitochondria inside of it, right? The DNA of the mitochondria is more similar to a completely random other alpha proteobacteria than the DNA in the mitochondria is in the cell, the DNA in the nucleus of the cell in which the mitochondria lives, okay? All right, so what is lateral gene transfer? Do you remember what transformation, transduction, and conjugation are? What's transformation? That's when, like, the bacteria picks up DNA off the ground like it's recycling it and then takes the bacteria and uses it. Yeah, that's transformation. What's transduction?
That's when, like, a bacteriophage virus goes and it takes the DNA from one place and puts it into a completely different other bacteria.
Yep. And what's conjugation?
That's when the two bacteria, like, they just make a tube and share DNA.
Yep. All right, so we think that genes from the endosymbiotic bacteria moved into the nuclear genome by lateral gene transfer. So we do find sometimes that we have some sequences in the mitochondria that are similar to sequences in the nuclear DNA, and we think that that was something that happened by transfer from the mitochondria into the DNA of the host. All right, so we also think that chloroplasts evolved by endosymbiosis, similarly to the way it happened with mitochondria. And so all of the photosynthetic protists have chloroplasts. And we think that that happened when a eukaryotic chloroplast, when a protist engulfed a cyanobacterium. So a cyanobacterium is a photosynthetic bacterium, and then a protist engulfed it. So we have evidence of endosymbiosis in chloroplasts. So chloroplasts, much like mitochondria, have bacterial-like characteristics. Chloroplasts have circular DNA with genes similar to those in cyanobacteria. Chloroplasts have peptidoglycan in their cell wall. And so remember peptidoglycan? Do you remember, when did we talk about peptidoglycan?
With the grandma stain.
I think it's a gram stain, not a grandma stain.
Did you know that peptidoglycan has dog in the middle of it?
No, I never noticed that before. Thank you for pointing that out.
Or dogly.
Okay. All right, so peptidoglycan, remember from the last chapter, is in the cell wall of chloroplasts, just like it's in the cell wall of bacteria. And there are living, extant means like alive in the world right now. We can find examples of endosymbiotic cyanobacteria that live in different kinds of protist and animal cells. So we can actually see this process in action. All right, so we think that there are two waves of endosymbiosis. So the first wave is called primary endosymbiosis. Okay, so we think that endosymbiosis first happened in the common ancestor of the Plantae, okay, the plants. And so at some point, endosymbiosis occurred in this lineage, and so all of these guys all have these chloroplasts, okay? Okay. All right, so that's a monophyletic group. There was a secondary endosymbiosis that happened afterwards. So chloroplasts can also occur in 4 of the other major protist lineages. And in those species, the chloroplasts are surrounded by more than 2 membranes, right? So it is possible that these guys engulfed one of these guys, or these guys engulfed one of these guys, or these guys engulfed one of these guys, which would explain the extra membranes. So let's just look at it up close here. So, no picture of primary endosymbiosis, I'll just tell you in words. So this is the endosymbiosis leading to photosynthesis in the common ancestor of all the Plantae. So you have a cell, and it basically eats some kind of cyanobacteria, and that happened in the ancestor of all the plants. Okay, there we go. And where did that guy go? Oh, he's right here. All right, so this is the result of the primary endosymbiosis. Okay? All right, what happens next? Well, some other cell engulfs this guy here that already has a chloroplast in him, okay? And so now you end up with a kind of a structure here. It eventually loses its own nucleus, and you're just left with a chloroplast. But instead of just having, so remember, the chloroplast was engulfed by this cell here, so it has its own membrane, and then the membrane it got when it went into this cell, right? Now this whole photosynthetic cell got engulfed by this other protist, and so we see here's the outside membrane, and then here's the membrane that it got from coming into this second protist, and then you can see they have 1, 2, 3, 4 membranes around the outside, and eventually this little guy loses its own nucleus. Okay. Okay, the nuclear envelope potentially also formed in a slightly similar way. We think that this happened because of infoldings of the endoplasmic reticulum. So rather than engulfing a particular thing, the membrane just pouches in on itself like this. And so it kind of pouches in, and eventually it encloses the chromosomes. Okay, and if you remember, when we learned about the cell and cell structure in 1113, right? The endoplasmic reticulum is directly around the outside of the nucleus. So if you have a strand of messenger RNA and it spools out of the nucleus, it goes directly into the endoplasmic reticulum. So this structure is congruent with what we see now. All right, so we can see that this makes sense as a hypothesis because the infoldings of the plasma membranes are also found in some bacteria that are alive right now. And the nuclear envelope and endoplasmic reticulum of the eukaryotes are continuous. So they're like one giant pouch, which you would expect if this is how they formed. Okay, so another thing, I gave you some reasons why eukaryotes are different than prokaryotes, right? One was the organelles, another one was the nuclear membrane, and then a third, here we go. So here's the nuclear envelope. So the nuclear envelope, well, I guess the previous slide explains how you could get the nuclear envelope from outpouchings, and then this slide is explaining its possible function, right? So having a nuclear envelope allows you to separate transcription from translation. So where does transcription happen?
In the nucleus.
Yeah, where's translation happen?
In the endoplasmic reticulum.
Yep. So there's lots of diversity in nuclei throughout the protists. So some of them have like two identical nuclei, like Giardia, look like little ghosts. Some of them have two differently sized nuclei, like a paramecium has a big one and a little one. Plasmodial slime molds, even though it has mold in the name, this is a protist, and they have nuclei that are like spread all around without a membrane separating them. So the cells don't have like dividers between them. Instead, it's just like each of these little glowy parts is like a nucleus, a nucleus, 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 down the length of the mold. What's a synapomorphy again?
It's a shared derived trait.
A shared derived trait?
Shared derived trait.
All right, thanks, Kat. What do you use it for?
Making phylogenies, I guess.
Yep. So we look for these different kinds of nuclear structures to be synapomorphies, right? And they help us to be able to distinguish the different protist lineages as monophyletic groups. The whole group of protists is not monophyletic, but each of the lineages is. All right, so protists vary widely in the kind of structures they use for support and protection. So lots of them have hard walls and external shells and kind of rigid structures inside the plasma membrane. But they can be very different in terms of what they're made out of. So if you look at a diatom, this picture doesn't really do it justice, but I'll show you a better one later. This shell is made of glass, so it's silicon dioxide. It's very beautiful. Dinoflagellates have cell walls that are made of cellulose. Foraminiferans have shells made of calcium carbonate, which are kind of chalky. Euglenoids, they don't have a shell at all. They have something called a pellicle, which is made out of protein strips, which gives them flexibility. So the evolution of all these different kinds of innovative structures for support and protection may have allowed the protists to diversify. All right, so we got to talk about multicellularity here because you start to see multicellularity happening in the protists. And when we say multicellularity, we mean that organisms that have more than one cell for an individual. I guess when you take when you take EEOB 3310 with me, we read some papers talking about the origins of multicellularity, but our best guess is that there were mutations that happened in cell lineages that made it so that, the cells are supposed to divide, right, and then separate from each other and go on and do their thing. But if you have a mutation that causes the cells to stick to each other after cell division, then you end up with cells kind of glumped together. And so one of our thoughts was that there may have been a mutation that caused this sticking, but then the cells that stuck together had some kind of a selective advantage that allowed them to reproduce more than the cells that were individual, okay? And so multicellularity has allowed these lineages to evolve and diversify. If you have enough different cells, eventually the cells can become specialized for different kinds of functions. So it's important to remember that every single cell, if they are made by, kind of just cells dividing in half, they should have almost exactly the same genes, right? But you can have the same DNA in all of your cells, but express different genes in different cells. And so we know that from Bio 1113, we've talked about it, right? So you have the same DNA in your eye and your liver, but the proteins that are expressed in your eye are going to be different than the proteins that are expressed in your liver to make, the structures that we need in the eye versus the liver. All right, so multicellularity did not just evolve one time. It arose independently in many different eukaryotic lineages. And I'll show you a tree of multicellularity in a minute. All right, so Protists have the ability to surround and ingest all sorts of things. They can eat little bits of organic debris, they can eat bacteria or archaea, they can eat other protists or even microscopic animals. So protists are not animals, right? But they could potentially eat an animal, you know, like a snail larvae or something like that. All right, so here's like an amoeba pseudopodia engulfing food. Other ones have these cilia here that kind of all beat together in the same direction and direct the food into the gullet where it is ingested. Yeah, so again, if you don't have a cell wall, you can ooze around and catch stuff. And this is a picture of pseudopodia, these long finger-like projections. So here's an amoeba attempting to eat a paramecium. So some protists actually actively hunt prey. Others will just attach themselves to a surface and then sweep food particles into their mouth with cilia. A lot of protists use absorptive feeding, and it means that they just kind of sit in an environment and take up nutrients across their plasma membrane. Other ones are decomposers, so they feed on detritus. Detritus is just dead organic matter. You have absorptive feeders that can live inside other organisms, and if it lives inside another organism and damages its host, it's called a parasite. You also have autotrophic protists. So what's an autotroph?
It makes its own ATP.
Yeah, it makes its own ATP. So it can make its own organic compounds by photosynthesis. They can range in size from just a single-celled photosynthetic protist to a giant seaweed. And for these guys, they're using carbon dioxide as their primary source of carbon. All right, so many protists you can actually see them behave. So if you turn a light on, they will be attracted to the light potentially if there's something that maybe feeds on the surface of the water. Or if you put food in, they can sense it and move towards the food. We have a name for the way a lot of them move. That's called amoeboid motion. So it's a sliding motion that is observed in some protists, like this one right here, where they stream their pseudopodia. This kind of a movement requires ATP. And in some ways, some of the molecular structures that allow them to move are similar to muscle movements in animals. And if you want to look at something that this kind of amoeboid movement is very similar to, if you look at our immune system, some of our cells actually use this same kind of amoeboid motion. All right, other kinds of protists swim using flagella or cilia. And so if you look at the structure of a flagella or cilia, structurally they're the same, but usually a flagella, you just have a couple of them, one or a couple of them, and they're long, and cilia are short and numerous all over. So the flagella will kind of beat, and then the cilia will kind of move in waves. It's not super useful to look at the kind of locomotion as a synapomorphia of a particular group, though, because we found that even protists that are very closely related to each other have very different ways that they move. All right, so protists can reproduce asexually, just by mitosis and cell division. So asexual reproduction, just as a reminder, is based on fission in bacteria and archaea, right, that are making genetically identical daughter cells. If you have sexual reproduction, we're referring to the fact that you have meiosis and fusion of gametes, okay? And in sexual reproduction, you're going to end up with daughter cells that are genetically different from their parents and genetically different from each other. All right, so name some advantages of sexual reproduction over asexual reproduction. Well, like if the environment changes, maybe you're more likely to have diverse offspring who can live. Yeah, that's one. What else?
Well, like if you have a lethal recessive allele, then maybe you'll get a normal one.
Oh, that's true. Can increase your genetic diversity.
Yeah.
All right, let's see. Name some advantages of asexual reproduction.
You don't have to find a mate.
That's true. Anything else?
You pass on all of your genes to your kids and not just half.
Yep. Okay. So meiosis is something that's adaptive for most eukaryotes because it allows you to have genetically variable offspring. And so if you have a change in the environment, then maybe you'll be lucky and happen to have offspring who have the combination of genes that will help them survive. So if we look at parasites and pathogens, if we kind of track their genomes are going to change very, very quickly, okay? And we know that this is because natural selection favors host individuals with new genotypes that might be able to resist these parasites and pathogens, and then the parasites and pathogens evolve in response to the host, and then the host evolves in response to the parasite and pathogens, etc. So you have this rapid evolution in both the host and the parasites as they try to outdo each other. So a lot of biologists will view sexual reproduction as an adaptation to fight disease. So if you are the host, right, you may reproduce only one time for maybe if your parasite is something little, it may be able to reproduce 100 times, right? When you only have one generation. And so if you have sexual reproduction, this is a way to come up with a completely novel combination of alleles that your parasite will not be able to deal with, okay? So sexual reproduction is a potential way that a host can fight a disease against a parasite that is able to evolve so quickly. Okay, so we're going to get into life cycles quite a bit, but let me tell you two different major types of life cycles: haploid-dominated and diploid-dominated life cycles. So when I talk about a life cycle, I just mean the sequence of events that happen as individuals grow, mature, and reproduce. And once meiosis occurred, which is something that's new in the lineage of Protists, this allowed there to be a distinction between the haploid and the diploid parts of the Protist life cycle. And so that would be different than something like a bacteria or an archaea. They are always haploid, right? Bacteria and archaea are never diploid, but eukaryotes can have part of their life cycle as haploid and part as diploid. All right, so I'm going to show you here a life cycle that's dominated by a haploid cell. So we're going to start out with a mature cell right here, and it may reproduce by mitosis, right? So it divides by binary fission in half. This is asexual reproduction. Its genetic material is exactly the same as the parent, and each of these two is the same as each other. And then they just, eat and get bigger, and then they grow until they become a mature cell again. So they start out as haploid, and they end up as haploid. All right, alternatively, this cell can undergo mitosis and divide into two smaller cells, like that. But then the two smaller cells can join up with each other, or maybe join up with a gamete from a completely different lineage. They fuse with each other. They form this kind of monster cell here that has two copies of each chromosome in the nucleus, so that's a zygote. Then it quickly undergoes meiosis to make two new cells that each have one set of each chromosome. These cells, though, are genetically different from the parent, right? And they're genetically different from each other. And then each of these will grow up. So for the sexual reproduction, you have fertilization, then meiosis, then growth. And for the asexual, you have mitosis and then more growth. Okay? So mitosis makes gametes, and then fertilization fuses these gametes. I should point out. So this is a life cycle dominated by haploid cells, so almost the entire cycle, whether it's asexual or sexual, they live as haploid, except for this little bit right here where they're diploid. Okay? This is a life cycle dominated by a diploid cell. So these cute little guys are little diatoms here, and this is what the mature cell looks like. In this case, the mature cell is diploid. It has two sets of chromosomes, okay? And so it could undergo mitosis and divide in half, kind of like a petri dish with a top half and a bottom half. And then each of these could kind of eat and then add material like this. When you end up with a really small one here, this can't undergo sexual reproduction. It's too small. It has to eat and grow up a little bit. But this one could, right? So here's your mature cell. So it can undergo meiosis. So it can make a cell that has half the number of chromosomes as itself. and this one has half the number of chromosomes. These can reproduce and produce a lineage that leads to either sperm or eggs. Then the sperm and the egg come together in fertilization. So now this is the brief haploid part of the cycle, back to being diploid. So this is diploid, 2 copies of each chromosome. This will grow up to become a zygote, and then this will keep growing to be a mature cell like this. Okay? So you can see most of the life cycle here is diploid with only a little short period of time as haploid. Alright, so this leads us to something that we call alternation of generations. And this is when one phase of the life cycle has a haploid form and the other phase has a diploid form. We call the multicellular haploid form a gametophyte. So it's the thing that makes the gametes. And so you have haploid gametes that are made by mitosis and then cell division. And if you bring 2 gametes together, you make a diploid zygote. So gamete plus gamete equals zygote. All right. At the other end of the life cycle, you have the sporophyte, which is a multicellular diploid form. So gametophyte is haploid, sporophyte is diploid. The sporophyte is basically the thing that makes the spores, right? So it makes diploid spores by meiosis. And then the spores over here will divide by mitosis to form these haploid gametophytes. All right, so this is a lot of words and a lot of vocabulary. So I'm going to summarize it for you in these I tried to make it like to memify it or something, or make it into a slogan. If we were here in class, I would make everybody say it 3 times for each line and just memorize this series of 5 statements, and it will help you not only for this section, but we're going to come back to life cycles over and over again when we go through the plants. And then when we go through angiosperm anatomy later in the 3rd unit. So remember this stuff, and you can remember it really easily just by memorizing this bit here. All right, so I'm going to have each of you guys say each of these 3 times, okay? And so we all know this is real dumb. Y'all can roll your eyes, ironically, but I encourage you, wherever you are listening to this thing, say it along with me. It'll help it stick in your brain, okay? All right, we ready?
Ready?
Are you going first?
Yes!
All right, go for it.
Gametophytes make gametes by mitosis.
All right, second time.
Gametophytes make gametes by mitosis. Gametophytes make gametes by mitosis.
All right, who's next?
I am! Gamete plus gamete equals zygote.
All right.
Gamete plus gamete equals zygote. Gamete plus gamete equals zygote.
All right, who's next?
It's me. Zygotes grow up to be sporophytes. Zygotes grow up to be sporophytes. Zygotes grow up to be sporophytes.
Who's left?
Sporophytes make spores by meiosis. Sporophytes make spores by meiosis. Sporophytes make spores by meiosis.
All right, I'll do the last one. Spores grow up to be gametophytes. Spores grow up to be gametophytes. Spores grow up to be gametophytes. All right, gametophytes make what?
Gametes.
Yeah, by?
Mitosis.
Yep. Gamete plus gamete equals?
Zygote.
Yep. Zygotes grow up to be?
Sporophytes.
Sporophytes make?
Spores.
By?
Meiosis.
All right, spores grow up to be?
Gametophytes.
All right, so I encourage you, whenever you get any kind of a cycle like this, just talk it through saying those slogans that we just said here, okay? So we're going to start out, gametophytes make gametes by mitosis, okay? Where's my gametophyte? All right, here's my gametophyte. Gametophytes make gametes by mitosis, right? Gametophytes are haploid. It doesn't need to undergo meiosis to make these gametes, right? It's already haploid. So gametophytes make gametes by mitosis. All right, gamete plus gamete equals?
Zygote.
Yeah. Zygotes grow up to be?
Sporophytes.
Yeah. What do sporophytes do?
They make spores by meiosis.
Yeah, sporophytes make spores by meiosis. So here, sporophyte undergoes meiosis to make these spores. Spores grow up to be?
Gametophytes.
Gametophytes. Okay? So this example here It's a weird one in which the sporophyte and the gametophyte look really similar, right? Okay, but we can still go understand what's going on if we just go through and say the things that we keep saying. All right, let's say the things again that we keep saying. All right, so where's our gametophyte? All right, here's our gametophyte. So what do gametophytes do?
Make gametes.
Yeah, how do they make gametes?
By mitosis.
Yeah, gametophytes make gametes by mitosis. There's the egg, there's the sperm. Then what?
Gamete plus gamete equals zygote.
Yeah, gamete plus gamete equals zygote. Zygotes
grow up to be sporophytes.
Yes, sporophytes make
spores by meiosis.
Spores grow up to be
gametophytes.
Okay. These are the 7 major lineages of protists. So the first one is the Amoebozoa. And so this is the lobose amoebos, means the one with lobes. This also includes the cellular slime molds and the plasmodial slime mold. So this and this are a cellular slime mold, and this one. So they basically kind of scrooch around in the ground as like these little cells, and then eventually when the food dries out, they send a signaling molecule to each other, and it causes them to all glob together into a giant slug. The slug will kind of slime around until it finds a good spot, and then it will form the stalk, and it will launch its spores off from the top of the stalk, and hopefully they'll land somewhere where there's more food. Okay, so that's a cellular slime mold. This is a plasmodial slime mold, so you might find something like this in your yard, and they grow really fast. These are the ones where they have like nuclei kind of spread around without a cell wall between them. Okay. Okay, so those are the Amoebozoa. Excavata are the ones that have that excavated feeding groove on the side, so you can kind of see it. Yeah, these are a good picture of it. On the side of this Euglena. These are Euglena. This group also includes the Giardia, right? These are the ones that can give you diarrhea if you drink swamp water. Trypanosoma, sleeping sickness, and Trichomonas vaginalis are 2 serious diseases, as well as Giardia. This is also the group that includes termite endosymbionts. So termites can eat wood because they have these endosymbiont protists living inside their gut that help them digest the wood. The next lineage is Plantae. Okay, so that's obviously the lineage that contains the plants, the land plants, but it also includes other things. This is ulva, or sea lettuce, volvox, which is like these colonial kinds of protists. Lots of these protists that we think are the ancestors of land plants. It also includes the red algae right here that look like this. And a lot of the different products that we eat contain different kinds of red or green algae, so obviously you can use it to wrap your sushi, but also we tend to use these kinds of algae as stabilizers and thickeners that you might find in things like toothpaste, candy, ice cream, lunch meats, yogurts, and gummies. Oh, by the way, so this is the Plantae. This used to be called the archaeplastids, if anyone has heard of that before in another class. The next lineage is the Rhizaria, and so that includes the foraminiferans and the others. So they don't have cell walls, but they have this shell-like covering, and they have these really little skinny pseudopodia. So instead of having lobe-like pseudopodia, they have these little rays that come out, and they have all these really cool shells like this, and like this, and like this one here. The next lineage is the Alveolata, and so this includes ciliates, meaning things with cilia all over the outside, like this and this. Dinoflagellates, like the one that cause a red tide. It also includes the group, this is the group that includes the malaria parasite and also Toxoplasma gondii. The next lineage is the Stramenopila, which is the one that we talked about at the beginning that had those weird the flagella with hollow projections. This is the group that includes the brown algae, which are these large multicellular organisms. It is the group that includes water molds. So you see mold, right? Don't assume that if something has mold in the title that it's a fungus, right? We already talked about cellular slime molds and plasmodial slime molds. In olden times, people would see something that looked like gross and they just call it a mold. But you know, if you've ever had the sad day where you come home and your fish has been engulfed in fuzz and is dead, this would be a Stramenopila protist that has attacked your fish, a water mold. And this group also includes the diatoms, which have these very beautiful glass shells, like this. And it is also the group that includes the protists that cause the Irish potato famine. Okay, so hopefully you learned all about protists and what kinds of themes there are in their diversification, the different lineages, different kinds of innovations in morphology, like the endosymbiosis theory and the evolution of multicellularity, and different kinds of innovations in feeding, movement, and reproduction.